Good morning, everyone. I'm Margaret Palmer, Director of SESYNC, and I want to welcome you to our seminar series. I'm super pleased today to let you know that our two speakers are postdoctoral fellows at SESYNC, and I'm going to introduce them separately right before each of their seminars. We're going to start with Dr. Katie Peterson. And she has an interesting background. She did her undergraduate degree at Louisiana State University. And from there, she went all the way across the US to Santa Barbara to go to probably the Bren School. And she was very much interested in looking at territorial use rights in fisheries. Then she went to James Cook University, so travels around the globe, and did her PhD, particularly focused on the use of ecological theory, the intersection of that with conservation biology. She's now doing a very interesting project at SESYNC focused on fisheries, and she's going to tell us about it now. Welcome, Katie.
Thanks, Margaret. So today I will be sharing with you guys my research on climate change and temperature variability in the Northeast Shelf regional ecosystem. First, I want to acknowledge the shooting that happened in Boulder yesterday. I'm based in Boulder now, and just want to say that it's unfortunate that there were 10 lives that were lost yesterday, and the US is really overdue for gun reform, whether that's a racist attack like last week or unknown motive like yesterday. And now I will attempt to talk about some fish. So the Northeast Shelf regional ecosystem is a large marine ecosystem located over the continental shelf of the Atlantic Ocean. It extends from Cape Hatteras in North Carolina to the Canadian Exclusive Economic Zone. There are 3 subregions that are entirely within US jurisdiction. It's managed by the New England and Mid-Atlantic Fisheries Management Councils, and it supports multiple commercial and recreational fisheries. The industrial sector employs 20% of commercial fishers in the US. Currently, harvested species are managed via catch limits that are informed by single species stock assessments, which are basically detailed models and assessments of one stock. NOAA is aiming to transition to ecosystem-based fisheries management by 2025, which aims to incorporate the ecological and social context of fisheries into management and policy. Part of this effort involves the parameterization of Atlantis for the Northeast Shelf. And Atlantis is an ecosystem model that incorporates all biotic and abiotic dynamics within the NSRE. So the NSRE has been experiencing some of the highest rates of ocean temperature change in the world. This is already impacting the fishing industry in New England. Climate change caused a decrease in employment in the industry by 16% from 1996 to 2017. Temperatures in the NSRE are changing 2 to 3 times faster than the global ocean average and are projected to get worse as the northerly Labrador current is predicted to retreat. And the warm waters of the Gulf Stream will then replace those by moving closer to the coast and further north. These oceanographic changes will result in shifts of suitable thermal habitats for some species. This figure shows that the thermal habitats within the NSRE for these 2 species, Atlantic cod and red hake, are projected to decline in suitability, which is represented by warmer colors. The warmer colors don't represent warmer temperatures here. Therefore, this region may no longer be suitable for some species, forcing distribution shifts out of the NSRE into Canadian waters. Similarly, some species that are currently managed by the South Atlantic Fishery Management Council may shift into the NSRE as they seek optimal thermal habitats as well. So because climate change is causing shifts in distributions of species, it's critical we think about climate impacts at a system level. Climate affects individuals through impacts in demographic rates. Higher temperatures are associated with higher rates of natural mortality or disease, and the timing and total reproductive output is affected by higher temperatures in many species as well. Additionally, metabolic rates are affected by temperature, which affects the satiation of predators. These all in turn affect the interactions between species, which have consequences for the structure of the community as a whole. With all of these dynamics occurring, it can be difficult to predict the net effects on the community and food web. This gets even more complicated when temperature variability is incorporated. As climate change worsens, the mean increase in temperature is usually accompanied by more extreme variability. So this brings me to my research questions. The first is, how does the projected abundance of a species change when temperature variability is incorporated into different demographic and metabolic rates? How do individual level changes and population level consequences due to temperature variability scale up to impact the food web? And finally, how can we plan for sustainable harvest that takes into account temperature variability? And does that differ from current management strategies? So as I mentioned, temperature variability complicates things. This is especially relevant in marine systems, as most marine species are ectotherms, which are cold-blooded organisms. And ectotherms generally have nonlinear responses to temperature. This is a thermal performance curve, which is one example of this nonlinear relationship. Temperature is on the x-axis, and the per capita growth rate, which is the number of individuals born to each individual, is on the y-axis. So if the temperature range is on the low end of the performance curve in constant temperature conditions, we would estimate per capita growth rate would correspond to this point in the curve. However, under variable temperatures, average per capita growth rate is higher than compared to constant temperatures due to Jensen's inequality, because this part of the thermal performance curve is a convex function. However, if an organism is living near its thermal optimum under constant temperature, we may expect the per capita growth rate to be at its maximum value. Under variable temperature, though, an organism's average growth rate would be lower because temperature is fluctuating such that the per capita growth rate is more often lower than this peak. It's not at the maximum. So species have different thermal performance curves in most cases. So let's look at what this means in terms of constant and variable temperature scenarios. Under constant temperature, we would expect species A to have its highest per capita growth rate, but species B would have one of its lower growth rates. Under variable temperatures, the predicted average per capita growth rate is slightly lower for species A, but slightly higher for species B. So is species A slightly less numerous in variable temperatures than in constant temperatures, and it would be worse off. But what if species A eats species B? If species A eats species B, species A may be better off in variable temperatures, as each individual is more likely to get something to eat because species B is more numerous, and species A has fewer conspecifics to compete with. So far, this has been looked at in pairwise comparisons of predation, but hasn't been scaled up to food webs, which is part of the aim of my work. This is a simplification of the food web in the NSRE. There are still a lot of interactions, as well as some cannibalism within species like cod and spiny dogfish. Fishing further complicates things because it changes the abundances of harvested species, and the relative abundances of species affect the strength of interactions between them. The loss of species as they shift out of the food web has the potential to rewire the food web, as some species may have to rely on other prey more or compete with other species for their preferred prey, or change the prey that they actually obtain. Right now, cod is one of the dominant predatory species in the NSRE. However, it's projected to have diminished overlap in its thermal habitat with multiple prey species, while spiny dogfish will increase in its overlap with prey species. Spiny dogfish may then become the dominant predator in the NSRE, changing the food web. When a new species shifts into the ecosystem, we can make inferences regarding which species it may interact with using link prediction methods based on traits or phylogeny, but there's still a lot of uncertainty around that. Fishing and restructuring the food webs due to climate change result in direct and indirect impacts on the abundance of species. And as I mentioned earlier, temperature variability has the potential to further impact the food web by causing changes in the magnitude of interactions between species. All of this has implications for fisheries. If the species that become more abundant are lower value, they aren't viable alternatives for fishers unless they can catch them in large volumes, which may not necessarily be sustainable. Additionally, the timing and seasonality of harvest for different species have implications for how fishers distribute their effort. So in marine food webs, size spectrum theory is an important explanation for the way they're structured. And it's also the foundation on which the modeling framework I'm using for my research is based. The size spectrum was first described following observations that smaller bodied organisms are far more numerous than large bodied organisms. In marine food webs especially, trophic position is impacted by body size. There are three fundamental scaling relationships to size spectrum theory. So first is prey preference. Larger fish eat smaller fish. There are other attributes that impact preference, and gut content analysis can reveal what prey was available and not necessarily preferred, but it does provide some evidence to support this relationship. Encounters between predators and prey are also impacted by body size. So here we have body size on the x-axis and clearance rate on the y-axis. Clearance rate is essentially the volume of water that can be searched during a given time period for prey. So a larger organism is able to clear or search more of the water better than and faster than smaller organisms. And finally, respiration and ingestion scale with body size as larger organisms have higher caloric demands. So MISR is a modeling framework I use for my research, and it is based on size spectrum theory for modeling marine communities. It allows for more complexity than a simple community model that may only look at birth and death rates and maybe interactions between species by incorporating explicit mechanisms that impact how populations change. However, it isn't so complex that it's impossible to parameterize. So that's why it's called medium complexity. It's more detailed regarding the community than a stock assessment, and it's not as intense as the Atlantis model that NOAA's working on. The functions within MISR can be modified and adjusted as well, so it's not a black box. The target number of species to include is around 10 to 15. Beyond 15, the model gets unwieldy, and this is because each species requires a data frame of life history parameters that can take up to 25 parameters. The species that aren't specified in the model are called the background resource, and this is generally the planktonic community and whatever else may be consumed by the focal species. MISR also includes a static interaction matrix that represents spatial overlap between species, and it can be modified to be dynamic through time. And finally, it allows for different harvest functions to be implemented within MISR to see how biomasses change. So I just want to highlight some work on different ecosystems that have evaluated climate effects using MISR. Most of the previous work incorporates climate via increasing temperatures based on IPCC representative concentration pathways. So not explicitly incorporating short-term temperature variability. Different metabolic functions have been modified using scaling functions in MISR. One is the Arrhenius scaling function, which is a standard scaling function that describes temperature increasing physiological rates. This has also been incorporated in natural background mortality rates in MISR. And finally, predator-prey interactions have been modified in the Hawaii pelagic system. Predator-prey interactions are thought to be improved by temperature to a degree by increased aerobic scope and forage success. The authors used a generic polynomial equation, which is mathematically a nonlinear relationship, which is essentially a thermal performance curve. But in this system, temperature was not explicitly varying. I'm building on this previous work by using MISR to evaluate 3 different scenarios. Changes to the food web due to novel interactions that arise following distribution shifts. Changes to the magnitude of interactions between species as thermal habitat overlap changes. And the direct impact of nonlinear responses to temperature and physiological processes. So as I had mentioned earlier, there are limitations to the number of species that I could include in the model. Too many species and it would never stabilize, too few and it wouldn't be representative of the food web. To decide which of the 80 species to include in the model, I've read through management plans, stock assessments, and have had discussions with NOAA scientists. And in terms of data needs, I needed to ensure that there was projections for their thermal habitats and time series data to fit the model. I wanted to make sure that there was a range of species with different economic values. So cod is a very important and iconic species commercially. Sand lance isn't harvested, but it's important in linking lower and upper trophic levels. And I also needed to ensure that there was some interaction at some point in time, either now or projected, as well as spatial overlap. And I wanted to make sure these species represented a range of biological and climate vulnerabilities. And these vulnerabilities are based on an assessment by John Hare et al. John Hare's at NOAA. And the biological vulnerabilities are attributes like being overfished, having specific habitat needs for spawning or recruitment, or having some other idiosyncratic life history attribute. Climate vulnerabilities describe how those biological vulnerabilities are exacerbated by climate change. So right now I'm getting my model running. Once that model is stable and all of my species are coexisting, I'm going to fit it to trawl and time series data from NOAA, and then I'll be able to incorporate the temperature effects and distribution shifts. So I'll have some sort of output like this. There are other ways to represent biomass, like through time as well. And then after my model is running and I have all of these scenarios accounted for, I plan on using the output for a thorough management strategy evaluation, as well as analyzing changes to the community by looking at different stability indices and the topology of the food web. So in summary, climate effects are important to consider within individuals, but we need to also scale up our evaluation to see how it flows on to affect community dynamics and food webs. Temperature variability is important, especially in ectotherms, due to the nonlinear responses to temperature. And finally, to make fisheries more sustainable, it's important that we have a mechanistic understanding of how climate affects the individual species and their abundances, as well as the repercussions for the food web, because that'll impact fisher behavior, and consequently, we need to manage for that. So I just want to acknowledge my colleagues that have been involved, my collaborating mentors, Laura and Kristen, the D Lab at CU Boulder, and my colleagues from JCU for being sounding boards for this work, the SESYNC community, Sarah Gacious, who has been really great in conversations to make sure that my work is relevant for NOAA and useful for them, the MISR size spectrum community for letting me pick their brain and learning more about the model. And I'd like to also acknowledge the Cheyenne and Arapaho peoples, the traditional custodians of the land on which I'm speaking to you today.
Thank you, Katie. Everyone I know is going to really look forward to seeing what the results are of the modeling outcome. If any of you have questions, please post them in the Q&A. But I will start by asking, Katie, are there examples or case studies from other very different kinds of systems in which the interactions have been taken into account as species shift?
Yeah.
So terrestrial system, freshwater, whatever.
There's been a lot of work on that. I have a colleague or friend, Alexa, who's based at Rutgers, and she's looking at that in the NSRE. Rebecca Selden's work, who I highlighted, has looked at that, and that's in a spatial context. There's definitely work that has looked at it, quantifying the interactions and changes. It's a very formative area of research right now.
I wasn't sure if that would provide any insights into some general patterns that communities are displaying in response to this shift.
Yeah, I think there's a lot. I can't speak to any papers off the top of my head, but I've definitely saved them and skimmed and read. So
yeah, so there's another question, and it touches a little bit on one I had also. Are there evolutionary responses on the part of the individual species with climate change?
yeah, and that's a really important thing to take into account, because some of these time horizons that are being modeled would allow for some sort of even intergenerational adaptation. There's a lot of work on that based out of JCU. I have friends that have looked at small turnaround times in fish, so ones that have short lifespans, and there's some indication that there's adaptation. I think that's really important to include and consider, but difficult to model.
Okay, a couple more questions. So Rachel Zurcher asks, acknowledging that you, of course, can't include everything in your model, what role does changing oxygen as the climate changes play in this system? Do you have any idea?
That's a really great question. And Elizabeth Dusky, who I mentioned on one of my slides, is actually looking at that right now in terms of the Baltic Sea. So I'm looking forward to seeing her research.
One last question. Were you able to talk to fishing communities directly or indirectly to get their opinions on key NSER species to the system?
I haven't done that. I think that would be great, but I've tried to take that into account in terms of reports from NOAA based on their discussions with fishing communities and fishers. I'm focused on the industrial sector, which is influenced a lot by how NOAA manages them, and they have town hall meetings often where they get feedback from fishers. So just the process of narrowing down to 13 species was time consuming in of itself.
Sure, complex project, and you're still in the early phases. So thanks very much, Katie. We really appreciated it.
Thank you.